Great. So, yeah, I am Keo Crubaugh. I'm a senior in biological and agricultural engineering at Texas A&M University, and my REU experience was in cave microbiology and the different tools we used for researching that. So, uh, Dr. Hazel Barton was my mentor, and I did the experience at University of Alabama. My field supervisor, who I did a lot of work with as well, was Max Kother. He was a PhD student in Dr. Hazel Barton's lab. As you can see, we're all expedition cavers, and our research revolves around cave microbiology. So, what is cave microbiology, you might ask? Deep within caves, even miles, even in the dark, isolated places, there is a microbiome, and those microbes do produce different byproducts through their metabolic pathways. Some of those things are like different acids, and those are used to maybe dissolve the cave walls, and different things like that. So while studying it, we can discover things about microorganisms, speleogenesis, which is like the creation of cave formations or the dissolution of like limestone to create the cave. Or sometimes it's like antibiotic resistance or drug discovery and just different chemicals that we can use in biotechnology. So that's what Dr. Barton's lab deals with. So the main project that I helped with was with Max Kother, and we were looking at these massive caves in Borneo. I didn't get to go to Borneo, but we just worked with the samples from there. But as you can see on the right side, that picture, these caves are like maybe 300 feet wide. They're massive. You see a tiny person in that picture, and they're wondering how these were created because nothing really makes sense. And they think it's from microbiology processes. So mainly dealing with the nitrogen cycle. So there's birds and bats that fly into this cave, really deep in this cave. Their guano is a energy source for bacteria, and those bacteria are producing different reagents, different acids that are potentially dissolving the cave walls, and like over a very long time have made these gigantic caves. We also did isotope ratio mass spectrometry to see the flow of nitrogen in the cave, and then we worked with some genomic extraction to see like how we could get the DNA to analyze out of this bird and bat guano because there's a lot of PCR inhibitors in there, and it's just really hard to work with. So that's a little qubit that we used to prove that the DNA was readable, and yeah, there's me doing preparing samples. So I also get to help out with Asurus silva. We were researching cyanobacteria in caves, specifically lava tubes, to kind of understand how life might survive in lava tubes on another planet like Mars. So helped him with some environmental sampling. I did some growth media preparation, helped a few other projects with that as well. Did some DNA extraction, and he showed me how he does sonication with the to extract the DNA. He had to work in this dark room with these green lights because cyanobacteria they use light for energy, so you can't really have that messing with experiments. Next, I got to go to the National Speleological Society National Convention. There, it was all cave research and science presentations. There was a lot of networking. My mentor, Dr. Barton, actually gave one of the main talks at the event, and most of the other PhD students in the labs gave talks as well. But I learned an insane amount of cave science, and then also got to do some caving trips while we were there because it was in Alabama or Tennessee, and yeah, it was great. So, one of the main projects I also helped with, apart from all the grad students, was my own thing. I was developing a procedure to test these microscopic rope fibers because they've been dissolving in this cave. So they're wondering why, why they're dissolving. That's a really scary thing to have your ropes dissolve in the cave. It can be dangerous. So I figured out how to manage these little microscopic ropes, treat them, and then use a tensile testing machine to test if they were breaking easier than the non-treated ones. It was really cool to develop my own procedure, and it's going to be used in the project for years to come. For science communications, I also made these little Instagram reels, as you can see there, and they did really good. Got a lot of good feedback on them, and did some podcast with all the grad students, which are on the YouTube channel. I'll have a QR code at the end if you want to check those out, and made a little survey like video on how to survey a cave with new technology. Other cave science, I was around cavers all the time, so they taught me how to do lidar analysis and actually search for caves using a computer and then go out and check them in person. Did some vertical techniques and also learned how to draft a cave map. So this is a cave I surveyed in Mexico, and they showed me how to draft it using Adobe Illustrator. In conclusion, the isotope ratio mass spectrometry showed good grouping. We could see how the nitrogen was flowing through the cave, gained new skills in cave science and microbiology, developed a nuanced method for genomic extraction from guano. We got really close on that. We got at least the DNA. It was proved that it was on its way to being extracted. They're still working on it. Developed a rope fiber testing procedure and gained a lot of experience in science communication. So I want to thank Dr. Barbara Gaston Wingenbach and Dr. Wingenbach and Dr. Leggett for the Science Communications program or Science Influencers program, and all the people I met at the University of Alabama that helped me along the way in different research and things like that, and my mentor, Dr. Barton. So that's it.